Hello everyone, my name is Aristea Binia. I'm a scientist at Nestle Research, and today I'm going to show you results from one of our observational cohort studies where we tested for associations between infant growth, adiposity, and HMO. HMO, or human milk oligosaccharides, we know that they are the third most abundant solid component in breast milk after lipids and lactose. We know that their concentration varies according to lactation stage and typically goes down over lactation with one or two exceptions. So this concentration can be from 5 to 15 grams per liter. We also know that more than 100 structures are present in nature, although less than 20 of them make the bulk basically in human milk. Most of the human milk oligosaccharides are actually not abundant in farmed animals. HMOs have been associated with several benefits or biological functions, typically beneficial modulation of microbiota, as well as protection against specific infection agents, or development of immune competence and protection from allergies. Now, the role in metabolic health and early growth is actually not so clear. What we have in the literature at the moment are a set of observational studies reporting individual associations between HMO and either infant weight or sometimes fat mass. And we also have some interesting preclinical studies demonstrating growth recovery in undernourished populations facilitated by sialylated oligosaccharides. And on the side from the clinical intervention studies, we know that the infant formula supplemented with either 2FL alone or 2FL and LNNT has no impact on infant growth. Typically, the baby's growth in the control arm as well as the experimental arm in these intervention studies grow in the same rate and following the WHO rates. Now, in our study, we sought to explore the associations of HMO with early growth, as well as adiposity, body composition, of predominantly breastfed infants in a multi-center European longitudinal study. Our observational study, our cohort study, was spanning in several countries in Europe. What we did is recruit women who intended to exclusively breastfeed. We measured their anthropometric characteristics as well as those of their infants in 6 time points. And for a subset, also we measured body composition by P-pod and collected milk at 2 days after birth, as well as 2 weeks after birth, 1 month, 2 months, 3 months, and 4 months after birth. Subsequently, we analyzed HMOs using a validated method and then tested for the associations of HMO between infant growth and body composition or secretor status again with infant growth and body composition across the 6 time points. The first observation we had was that the infants in this population grow within the normal range according to the WHO growth charts, and we found no difference by secretor status. In the plots that you see, we have weight length as well as weight for length for male and females, and the white box plots are the non-secretor mothers, so babies fed by non-secretor mothers, and the black babies fed by secretor mothers. So as you see, they grew within the range of WHO depicted by the percentiles shown in the figure. We also looked at fat mass and fat mass index during these 4 first months of life, and we did not find any significant association by secretor status. We also use principal component analysis to identify any relationship between fat mass area under the curve or fat mass index during these 4 months of lactation and individual HMO. As you see in the 2 plots, the first one on fat mass and the second on fat mass index shown in tertiles, we identify no specific cluster of HMO by fat mass or fat mass index. We did identify, however, modest correlations with individual HMOs and infant growth and adiposity. First, at 4 months, we saw that the percentage fat mass was inversely correlated with LDFT and positively correlated with MFLNH3. At 1 month of age, infant weight was inversely correlated with 2 sialylated HMO, LSTC as well as TSLNT. And finally, at 4 months of lactation, infant weight was inversely correlated with 3SL. We also tested for associations with HMO using a regression model. And in that case, we modeled fat mass accretion association with individual HMO, and after adjustment with for pre-pregnancy BMI, sex, birth weight, as well as fat mass at visit 1, we only identified 1 single HMO significantly associated with fat mass accretion. In that case was again MFLNH3, as we found for percentage fat mass at 4 months of lactation. In conclusion, we only identified modest associations or correlations between growth and adiposity in this population. Our population was a population of healthy growing infants, exclusively or predominantly breastfed by well-nourished mothers living in Europe. HMO, however, may modulate the risk to future metabolic disease. What we would propose for the future population studies is to focus on groups or profiles of HMOs rather than individual HMOs, and address this association with either growth or adiposity in the context of specific healthy disease, and as well as apply a long-term follow-up to assess the impact on the future development of metabolic disease. I would like to thank all the investigators and the collaborators of our observational study, the Atlas of Human Milk Nutrient and Multicenter Observational Cohort Study. Thank you very much.